Hello, my name is Sion, and I am an assistant instructor for the Georgia Tech School of Biological Sciences, and this is a review of the genetics unit for Biological Principles 1, so Intro Bio. And the thing about the genetics unit is it's a shortened and condensed version of basically a semester-long genetics course. And so what I've done a little bit differently with this review session is that I've split it up along the natural division of where a genetics course covers material. So this first video on genetics 1 is going to cover more of the inheritance and actual genetic side of things, whereas genetics 2 will cover DNA and DNA structure. So with that being said, let's go ahead and get started. I've also updated the format of these slides a little bit, so I have a few tips on how to best use them. As an update from last semester, what I've done is I've taken my slides and I've actually removed the learning objective from my copy of the slides, but what I did was in the description, I put a skeletal version of the slides that have the learning objective on them because we should cover every single learning objective from the lecture with this review session. And I think it would be really good practice. I'm a big believer in using active recall, and I'm a big believer in being able to take information and write it in your own words. So with the handout, that's a really good resource to take the information I'm presenting to you and choose what you think is important or what you don't know and put them on there. And then a big thing was that in 2025, the study came out and it said that listening to lo-fi does help when you're studying. Obviously, I can't tell you what to do, but if you want to do that, it works. That's why I listen to Subway. That's why I play Subway Surfers in class. So whatever works for you, it's been proven. So really good luck locking in, and let's go ahead and start. So the first thing we have to start out with is some basic vocabulary, just because genetics has so many terms and it's really important we get those straightened out so that way we know what we're talking about. So obviously the first term is going to be whenever we talk about a chromosome, we mean the structure consisting of DNA and protein that is carrying genetic information in an organism. And so obviously, if we have a chromosome, a big thing that you hear in this unit is the word homologous chromosome. So what does that mean? Well, homologous, right? Think homo, think same, think good notes is not good at having a good highlight, but homologous, same. So what that means is that we have two chromatids that have come from each parent and they have the same genes in them. So they're homologous. They are the same on both sides. So like an X structure like this, right? That is a homologous chromosome, right? And so that's this as a whole. And so each individual part of that is a homolog. And when we have two homologs, because they're homologous, they're the same, that's how we get a homologous chromosome. They're not going to give you like a microscope slide and you have to identify a homologous chromosome, but it's important to know what we're talking about. And that also lets us talk about the state of replication of a chromosome. So chromosomes can either be replicated or unreplicated. Obviously, when we look at up here, where it says haploid, really that's unreplicated, right? Where we have just the one. When we become replicated, and I'll write that down here, we have an identical copy where we have two of these, where we get, that's our X, where they come together, right? So that's where we're talking about unreplicated chromosomes, and then also sister chromatids. That's what that individual part is called, where when we have the homolog and it comes together to form, right? We have the homolog and it comes together to form a homologous chromosome. We have two sister chromatids. And that gives us a natural segue into being able to talk about the chromosomal content of a cell in terms of ploidy. So when we have a cell, the chromosomes can either be unreplicated or replicated. And what that means is that gives us a way in describing the chromosomal content based on how many sets of chromosomes are available, right? So we can have either one set or we can have two sets. And if we have two sets, that's going to be diploid. And if we have one set, that's going to be haploid. And for both one set and two sets, there are two different ways to be diploid or haploid. So for example, we can have one set of chromosomes consisting of just one sister chromosome. So because we have one set, that means that we're haploid, and this is going to be one unreplicated. Okay, let me figure out how to write that better. Sorry, there's not a lot of space in that box. So we have one unreplicated. Well, what happens if we make an identical copy of that, and that's where we get that X structure? Well, when we copy that, that's when we have two sister chromatids that are replicated. We're still haploid because we only have one set of chromosomes in total, but now it's replicated because we have the same thing on both sides. Same idea, how can we be diploid? Well, if we have two different chromosomes, right, then we're diploid because we have two sets. But they can consist of either two separate sister chromatids, or they can, again, become replicated. And that's where we would have four sister chromatids. See, even I get tripped up with the vocab. But basically, whenever we have two sets of chromosomes, that's when we're diploid, and whenever we have one set of chromosomes, that's when we're haploid. And they can either be replicated or unreplicated. And I can almost guarantee you there's going to be a question on the exam where it tells you what you see in terms of chromosomes inside a cell, and you have to determine if it's haploid or diploid. That's why I have this practice question right here. So let's look at it. Again, we're examining cells. So you might wonder how I know there's going to be a question about this on this exam, because I took it from a past exam. But we're told that in the cells we have two sets of chromosomes, and each chromosome consists of two sister chromatids. And then we have another cell that has one set of chromosomes, and each chromosome is composed of one sister chromatid. So really, what you can do with this question is, if you really want to, you can just draw this out so you can have the answer. So for our first cell, we have two sets of chromosomes, okay? And each chromosome, so 1, 2, and each chromosome consists of two sister chromatids. So boom, boom. What about with our second cell? Well, we only have one set of chromosomes, and it's composed of one sister chromatid. So let's go ahead, not even look at the answer choices. Let's just identify this for ourselves so we know. Well, if we look at the first cell, we have two sets. So we know right off the bat, hey, it's diploid. But then we look and we say, hey, there's two sister chromatids in each chromosome. So we know that they've been replicated. What about in our second cell? Well, we have one set, so hey, that's haploid. And we only have one sister chromatid, so it's unreplicated. So we can just go ahead and choose the answer choice that matches with that. And that's going to be answer choice B. And again, they're going to ask you about this on the exam, so I know, just draw it out. That's the best advice I have for this type of question. So now let's start with how cells grow and divide. So there's two different ways cells do this. We have the process of mitosis and we have the process of meiosis. Okay? And I'm just going to list the key points about this because they're not going to really get into the details of what these processes do. But mitosis, right, produces genetically identical cells. That's the big idea. And they're produced asexually. Whereas meiosis requires, is performed through sexual reproduction. So the combination of sperm and egg to produce genetically different cells, aka gametes. So with mitosis, we go from 2N to 2N, and that new 2N cell is identical. And with meiosis, we now go from 2N to N, and this cell is going to be genetically different, this gamete. And that's a very important process, and that's how we get genetic diversity. And you can kind of see that graphically here, where with mitosis, we produce two daughter cells that are genetically identical, and they are both diploid. However, with meiosis, we go through two different divisions, and we get 4 haploid daughter cells. And as for the stages of this process, the acronym for that we've all been taught is PMAT. So prophase, metaphase, anaphase, telophase, and that just describes how the how the chromosomes are moving in the cells, where they align and then they are pulled apart, and that happens one time in mitosis, and that happens twice in meiosis. And here's a graphic of that again, and that also tells us a little bit more about the chromosomal behavior in the cells. So in mitosis, basically, we just have the chromosomes align on the metaphase plate, and then they are pulled apart during anaphase. In meiosis, the important thing is that what this is saying right here, in this whole paragraph, this is basically saying that crossing over occurs, right? Where we have that exchange of genetic information between two of the homologous chromosomes, right? That's what it's saying right here. And so what that ends up happening is that when those chromosomes are pulled apart, we see here that after the first division, these daughter cells are going to become haploid because they're only going to have one set of chromosomes. And you can see that here, simpler, where after the first division we have our 1N cells. And then after anaphase 2, those the replicated chromosome gets split into just the sister chromatids, which is how we get 4 cells, each containing one sister chromatid. Big idea here is nondisjunction. So this happens during meiosis when either the homologous chromosome or the sister chromatids cannot or fail to separate during meiosis 1. So that's what's supposed to happen, and what happens when that process doesn't occur as it should? Well, that's called nondisjunction, which is the failure of chromosomes to separate properly during meiosis. So that's either going to happen when the homologous chromosomes fail to separate, or their individual sister chromatids. Okay? And that can either happen during meiosis 1, which is when the homologous chromosomes are supposed to separate, or meiosis 2, where the sister chromatids are supposed to separate. It can also occur in mitosis sometimes, but that's obviously not going to be passed to any offspring because mitosis produces somatic body cells and not cells involved in sexual reproduction. And that's how we get, like cancer cells, which have an abnormal chromosome number. But we see here graphically where that happens, where we have non-disjunction with this chromosome. So we end up with one daughter cell that has 3 and one daughter cell that has 1. So we get this weird N+1, N-1 situation in our 4 daughter cells. Because some have an extra sister chromatid and some have one less than they should. So again, we see here where we have this question about non-disjunction. And so really when we're looking at the answer choices, we have to identify what must be true, right? And when we look at these, right, we see, 5 chromosomes in the gamete, 10 chromosomes during metaphase 2 of meiosis 2, 5 homologous pairs in metaphase 1. And the thing here is that if non-disjunction happens, we have no idea what happens to the chromosome number. We are not told. So the only thing that can be true is that it's going to have a different number of chromosomes. That is the one guarantee that will happen during non-disjunction. The rest, we have no way of knowing unless the question provided us with more information. Now, this is how cells grow and divide, which is called the cell cycle. So different stages of that. So the cell cycle starts with the G1 phase, aka the growth phase, where the cell is growing and it's preparing for DNA replication by ensuring during the G1 checkpoint that the conditions are right to replicate and become another cell. Then we have the S phase for synthesis, which is when DNA replication actually occurs. So big thing here, and you can see this on a graph, which we will talk about, but the genetic material in the cell is doubled. Okay? Sorry, there we go. And then we have the G2 phase, where the cell then prepares for mitosis. And then lastly, we have the M phase, which is the actual splitting of the cell, and that's where we see my acronym come back, PMAT. And that's the process for which the cell divides. So here's where things can go wrong in the cell cycle, and you have another graphic depicting what happens in the cell cycle. So obviously, if during the G1 checkpoint the cell does not properly regulate when it should start dividing, that's when we get cancer, because the cell will divide uncontrollably, right? Other times when we're checking to make sure that the cell has done the right thing, during the G2 phase or the S phase with our DNA, replicating it correctly, that's when we can say, hey, this is not right. Let's just kill the cell off. That's called apoptosis. If that doesn't happen correctly, that's when you would lose tissues or other cells because the cell would have incorrectly died or been killed off. Big thing here is aneuploidy, which is an abnormal chromosome number, right? That's basically like when non-disjunction happens during the M phase, right? The M phase is when non-disjunction would happen. So now we're asked about the DNA content of the cell during various stages. In this case, we're asked when a cell has the most DNA during meiotic replication. And remember, meiosis brings us from a 2N cell to a 1N haploid cell. And so that means that whenever there's going to be the most DNA, it's going to occur way before that division. It's going to occur when the DNA has been replicated. So it's going to be all the way before here. And when does that happen? Well, what we're really looking for is when the DNA has been replicated, because then we have two copies of it, right? AKA the synthesis phase. So let's look at the answer choices. Where does the synthesis phase occur? And we can rule out basically everything based on that, identifying that the DNA content in the cell is going to be greatest when we synthesize a copy of it, right? So it's not going to be before replication. That makes no sense. It's not going to be during interphase because that's has a normal amount of DNA. And it's not going to be in meiosis 2 because the cell is already divided. And not after two cells form because, again, every division reduces DNA content. It's going to be between interphase and division, right? Let me even write that out because that's a really key idea. Between interphase, and the actual division of the cell, we have to have replication, or the synthesis of a new DNA. And that's going to occur before the first cell division. So now what we can do is we can look at the DNA content in the cell over time in the form of a graph. And so this graph can show both mitosis and meiosis. So the best way to interpret this graph, in my opinion, is to think about the different outcomes between mitosis and meiosis. And what do we start with? Well, in both mitosis and meiosis, before the cell divides, the first step is we just have our regular cell. And then as it prepares for division, that is a horrible circle, as it prepares for division, it has to make an entire copy of its DNA. So the DNA in the cell doubles. And what phase is that? That's the S phase. And that's where we actually get the differentiation between mitosis and meiosis. So mitosis, when we divide, we take this cell and we just split that DNA between two daughter cells, right? So we have 2N daughter cells, right? So we went from 4x the DNA content down to 2 times the DNA content because that 4x was split between two different cells. Now with meiosis, we know that we go through two divisions. So in the first, we have 2N daughter cells, and then each of those produces a daughter cell with one sister chromatid. So we go from having 4x the DNA content to 2x down to just 1x in each of the cells, right? And now we can go back using these outcomes and put that in terms of what's happening to the DNA content with the graph, right? So we know that we start with 2 times the amount of DNA in the cell, then we replicate it, and that's during the S phase, so we end up with 4 times the DNA content, and then we go through a division. And this division brings us back down to 2 times the DNA content. So this division will either be mitosis or meiosis 1, right? And if you think about it, it makes a lot of sense that we go from 4x to 2x in a cell because we have 4 times the DNA and we're splitting that between two of the daughter cells. So it's 2x in each. And if this is meiosis 1, that's where cells become haploid. That's an important thing to recognize. And obviously in mitosis, they stay diploid. And then if we go through another division, what do we end up with? Well, we end up with 1 times the DNA content, and that's going to be meiosis 2. And I think understanding this graph is a really important thing to be able to do. Think about what is happening to the chromosomes in the cell, right? Because first they get replicated, and then they get split into daughter cells. So be able to recognize what's happening to that DNA content. You can even just memorize this graph, memorize the annotations I put on there. And I would do that because if I was a betting person, I would guarantee that there is going to be a question about this on the exam, right? And so what ends up happening? Well, in this question, we're asked, hey, when do cells become haploid and when do they have only a single sister chromatid? Well, we know, if we recall the graph, a single sister chromatid, that's 1 times the amount of DNA. And where do we get that on this graph? Well, we get that at step 4, where we have 1 times the amount of DNA. And then when do cells become haploid? Well, if you recall what's happening, right, that's basically saying that we have the DNA split between 2 cells. So we have a 4x and we go to a 2x. And at what point does that happen? That happens right here where we go from our 4x in the S phase down to 2x in our 2 daughter cells, and that's going to happen at 3. So the correct answer is stage 3 and stage 4. So again, really important to be able to recognize what is going on with this graph, what is going on inside the cell. Now we get to talk about Mendel and him playing around in the dirt with his pea plants, right? So Mendel discovered, by playing with his peas, 2 of the most fundamental laws of how alleles are inherited. So the first law that he discovered was the law of segregation. And what the law of segregation is basically saying that 2 alleles for each trait, so in this example, Y, are separated during gamete formation. So they're segregated, they're separated. So each gamete receives only 1 allele from each parent. That's what the law of segregation is saying, right? So once our parent cells, once they go through that division process, right, we have the chromosome, the X, split and move to opposite sides, which means that when we divide and create daughter cells, the daughter cells are only going to receive 1 allele from each parent. And that's where we see here where the P generation stands for the generation of the parents, where the parents have 2 alleles for each trait, big Y and big Y, and little y and little y. And because of the law of segregation, the gametes produced will only have 1 from each parent and produce offspring with the big Y from one parent and the little y from the other. And that's what the law of segregation establishes. The second idea that Mendel established is the law of independent assortment. And he discovered this by looking at his peas, where he had 2 traits, right? We have round and we have wrinkled. So that's the physical appearance. And we have color. We have yellow and green. So we have the appearance, round and wrinkled, and we have the color. And he discovered that one trait is not going to influence the inheritance of another. This happens completely separately or completely independently of one another. So what the law of independent assortment is saying, and right here, that's the big idea. It's saying that alleles for different traits are distributed to the gametes independently of one another. Now we're just supposed to learn how to draw a Punnett square, and this is based on the principles of Mendelian inheritance. So let's do a Punnett square for, let's say, a cross between yellow peas and yellow peas, and they're both heterozygous. So we have a capital Y and a little y, okay? So on the top, let's put big Y, little y, big Y, little y, and then we're just able to go across. So we got big Y, big Y, big Y, little y, big Y, little y, and little y, little y. So here in the top, we have homozygous dominant, and across from it we have homozygous recessive, because we have 2 copies of big Y, which is dominant, and we have 2 copies of little y, which is recessive. And then in this diagonal, we have 2 heterozygotes, where we have 1 copy of big Y and 1 copy of little y. Now we can do the same thing, but in a dihybrid cross, where we've added on a second trait. So let's say we again have our heterozygous yellow plants, but we also want to add on the trait of roundness or shape. So we have big Y, and then we have big R, little r. So when we go ahead to do this cross, we're going to start with YR. So big Y, big R, big Y, little y, big R, and little y, little r. And we do this on both sides because that's how we would want to distribute the two different traits. And those are all the different possibilities. So then when we go ahead and we fill in this chart, we have the different homozygotes and heterozygotes, and we see them distribute themselves, and I've done the different highlights for this, but we see them distribute themselves in a 9 to 3 to 3 to 1 phenotypic ratio, where we have 9 that are yellow and round, and 1 that is green and smooth, or whatever the homozygote recessive is, so I guess wrinkled. That's why would you use R for wrinkled? I mean, seriously. And then we have the 3, yellow and wrinkled, and we have the 3 green and round. So yeah, it should come out in a 9 to 3 to 3 to 1 phenotypic ratio. And we see this here with a graphic, which makes me think why I even wrote that out. But yeah, we have 9 smooth and yellow, 3 smooth and green, 3 wrinkled in yellow, and 1 wrinkled in green. And you can see that here where we have the different colors depicting that. So now what we can do is we can use these Punnett squares and apply some basic mathematical rules in order to calculate the probabilities of receiving a specific genotype using a formula, right? So we know that if we have a monohybrid cross, we have a 3 to 1 chance of being dominant to recessive, and we have the 9 to 3 to 3 to 1 ratio in our dihybrid cross. And if we recall, there's this idea, each parent is only going to contribute 1 allele per gene. And that's the law of segregation. So with an example, let's say we have the trait big A, or the alleles big A and little a, right? And we want to look at the probability that we end up with the homozygous recessive little a, little a. So parent A has a 0.5% chance of contributing the little a allele, right? Because we have 0.5 of contributing big A, 0.5 of contributing little a. And what about parent 2? Well, we're going to multiply that by the chance of getting little a again, which just happens to be 0.5 again. So we multiply these together and we get 0.25. So we have a 25% chance of being homozygous recessive. And if you look back at a Punnett square, obviously, hey, that makes a lot of sense. That's where we get our 3 to 1 ratio from. And that's why it's really hard to come up with a reason to use the probability rule in this class. But the idea is that for each specific event, you would multiply them together. And if the events are exclusive of each other, you can add them together. And that's the sum rule. So for example, the probability of getting a big A or the probability of getting little a, and that's when you could take 0.5 plus 0.5. And the probability of getting either of those is literally going to be 1, which makes a lot of sense because, I mean, look, you have a 50% chance of getting either. And now we can talk about the units of inheritance, so our chromosomes and our genes. So now we get to talk about Drosophila, which are fruit flies, and you will hear this come up in every genetics class ever because they established the idea of sex linkage. So these flies, they have 2 potential traits, right? They have eye color, so either white or red, and they can either be male or female. So that's trait 1, and trait 2 is their sex. So what happened when he crossed them together was that the females all had red eyes and the males all had white eyes. And that was really weird because that was like 2 traits were coming together, which shouldn't happen because that would violate Mendel's law of independent assortment. So this led to the idea that, hey, somehow sex and eye color are linked. Okay? And that makes a lot of sense when you think about it because males only have one X, which means that whatever version of the gene is on the X chromosome, it's going to come across. But females have 2 X's, which means that even if one of them has the allele for white eyes, right? Even if one of them has the allele for it, unless the other one does, it's not going to show that, which means that, hey, the eye color trait is on the X chromosome, which means that some traits can be sex-linked. And that's basically what this practice question is asking. Hey, we're told that these two traits do not assort independently, they're inherited together. How can this possibly make sense when we know that traits should be coming over on chromosomes? Well, if we look at these answer choices, some of them are just blatantly wrong, and we have to pick out the one that makes sense, right? Well, if we look at these answer choices here, some of them are just straight up wrong, right? Genes assort independently because they're always inherited separately. Is that true? Is linkage been disproved? No, so we know it's not A. Traits are inherited independently of chromosomes. Well, I read that and I just stopped reading. We know that's not true. The inheritance of traits is controlled solely by dominant and recessive alleles. No, because we just saw sex linkage, which has nothing to do with dominance and recessive, which means that genes on the same chromosome can be inherited together, and we have the homologous recombination during meiosis that gives us genetic diversity. So the answer is B. And again, that's where we see our ratios come back, like when we talked about probability, where with a monohybrid cross, we would see a 3-to-1 ratio, and a dihybrid cross, we would see a 9-to-3 to 3-to-1 ratio, right? And if something deviates from that, because this is what is expected by Mendel's P's and his experiments, this is what you should get. If something deviates from that, that would infer the presence of linkage. So remember those two numbers, those are like the magic numbers for this class. And now we get to talk about pedigrees. Yay! Right? So pedigrees show how traits move down generations. And there are 3 types of pedigrees you will see in this class. And I'm just going to go ahead and tell you, for advice, just memorize the patterns. Pedigrees are based on your ability to see and recognize a pattern because there are only 3 different types. So the first type is autosomal dominant, which is just a trait where the trait is passed from one parent to the children. And because it's dominant, you only need 1 copy. So you literally have a 50-50 chance of getting it. What about autosomal recessive? Well, think what a recessive trait is. You can pass it down, but it requires 2 copies. That's what it says right here, right? It requires 2 copies of it, which means not only can it skip generations, but it's only going to affect 25%. And lastly is X-linked, which should primarily only affect males. Why? Because males only have 1 X, so whatever version of the X they inherit, they're going to express that gene, right? And that means that daughters can carry it because, for example, in a male, if you have X with the bad gene Y, you're going to express it. But in a female, you can have an X with the bad gene and then X without it, and you're going to be a carrier. So like I said, I would just recommend memorizing the different types of pedigrees that can appear. You see here with autosomal dominant, hey, both parents have it, so you get offspring that have it, and then just to continue the pedigree, one by chance doesn't have it, and those two can come together and make offspring without it. But then again, it's going to appear no matter what, and at least one of the offspring. It cannot skip a generation like that. Autosomal recessive, we see here that you only have a 25% chance of getting it. Literally, we had 4 offspring, 1 had it. We have 4 offspring, 2 had it, which means that this guy must have been a carrier for it. Again, seriously, just memorize, yeah, because that's how you would get that. But just memorize what these pedigrees look like. That's my best advice. And right here is X-linked. How do I know that? None of the daughters have it, only the males, which means that we must have a carrier, or this must have the bad allele, either one. You can kind of easily distinguish the differences between the pedigrees. Okay, so here we have a practice question about an X-linked disease. So if the father has the X-linked disease, I will literally write X, bad, Y, and the mother does not. What is true about their sons? Okay, it's X-linked and it's recessive. So the mother, if the mother had it, it would literally have to be X bad, X bad. But the mother doesn't. So the mother can either be X bad X or regular XX. No matter what, this means that when we get a son, we're crossing X bad Y with either X bad X or XX. Which means that either half of their sons will be affected, right? Because the sons only need one copy of that X. Or half of them will be completely unaffected because they will get 2 copies of a regular X. And so, again, we know that the father, because he is expressing it, has a copy of the bad allele. And the mother, we're going to cross that with either having the bad allele and not, or not having it. Okay? And that means that either none of them will be affected because you'll inherit a non-bad X, or half of them will be affected because you'll inherit a copy of the bad X, which makes A the correct answer, even though it's bolded wrong on my answer key on my slides here. And that brings us to the idea of linkage, right? We talked about this where we can have 2 traits on the X, the sex chromosome. Well, linkage overall just happens when 2 traits are very close to each other on the same chromosome. And this means that when crossing over occurs, just by random chance, sometimes these will come over together. And that means that this is going to violate the law of independent assortment, right? Because it means that 2 traits came together instead of assorting independently. Okay? And linkage is going to cause a deviation from Mendel's ratios. So either 3-to-1 or 9-to-3 to 3-to-1, which is why I said these are the magic numbers. Again, practice question on this. Hopefully you should be able to get this. I don't think I need to talk it through. The correct answer is just 2 because I literally never mentioned segregation, and it's also 2 genes can be on the same chromosome. They don't have to be linked. They can be far away, right? This B trait could be all the way down here, and that would mean it's far enough away that it's likely not linked. Okay, moving on. Again, now we get to talk about patterns of inheritance. And the big idea is that these patterns, well, they exist in a perfect world, right? Well, truthfully, that's not what happens with a lot of traits, and that's where we get different patterns of inheritance. And that's what this slide is basically acknowledging. Before we can talk about the different patterns, I want you to recall that gene expression and phenotypic expression are different things. So, phenotypic expression, I will say, that's like being physically expressed, where gene expression is happening on the DNA level. For example, we have this dominant pink allele. So just having one copy of it creates a pink flamingo. However, this pink flamingo came from a purple flamingo and a pink flamingo. The genes coding for purple are still expressed. That's very important to understand. Gene expression is occurring. The purple protein is getting made, but is being masked by the pink allele because it is dominant. Right, we see here, this is a practice question talking about the difference between phenotypic expression and gene expression, right? Butterflies produce the same dark pigment when the gene is active. However, in lower temperatures, we become lighter. So what is the difference between the expression of the genotype and the phenotype? Well, we know that these butterflies have the same genotype, but something is happening to make them lighter. And the lighter color, that is a change in what? The genotype or the phenotype? That's a change in the phenotype. Okay? So that means that we have to look for the answer that says, hey, the genotype is coming across, but the phenotype is not coming across because of the lower temperature. Something going on with the environment. And the answer choice that matches that is answer choice A. Okay. So there are 3 different types of traits, right? And this is acknowledging, hey, stuff deviates from Mendel in what he's saying. So we have quantitative traits that have continuous variation, right? So for example, height or skin color, where we have a distribution, right? And you see that in like a bell curve, right? Where height varies, skin color varies, people's blood pressure varies, and that's going to be influenced by multiple genes. Multiple allelism occurs when there's more than two alleles for a trait. Biggest example of this is blood type, right? So you can have type A blood, type B blood, or type O blood, where we have two little i's. But the idea is that there are more than two alleles for it. And polygenic inheritance is when we have a trait, our phenotype, being controlled by multiple genes, right? So skin color comes from how much melanin we're being produced, and that's controlled by multiple different genes, multiple alleles coding for that. Now, in terms of how things can be inherited, we have epistasis, aka gene by gene, where one gene is going to modify or mask the expression of another, and you have an example of that there, or gene by environment, which hopefully you can recognize this, where the gene, the phenotype, gene by environment, and we have gene by environment interaction, which changes the phenotype, where it's going to be influenced by not only the alleles, your genes, but what's going on in the environment. And you have another example of that there. So we have this practice question here that's basically asking about that pattern of inheritance, right? So we're told that we have two genes, A and B, that determine flower color. A produces a red pigment, but only if B is present. Without B, the genotype will be A. Well, let's go ahead and recognize what this is saying. So what we could do here is we can put this question in a little bit simpler language, and which is A requires B, or B modifies A. And hey, that sounds a lot like epistasis, which is a gene by gene interaction. And that's the correct answer. And there are also different types of dominance. We have incomplete dominance and codominance. And hopefully you've seen this in high school biology. But the idea is that incomplete dominance, the idea here is that in incomplete dominance, even though you have a dominant allele, it's not going to come across. You're going to have an intermediate or a blended trait where the dominant allele is not completely expressed. That's where we would see this, where we have this pink flower instead of a completely red flower. In codominance, that's when both are expressed. They're codominant. They come across together. We have a red flower and a white flower, and that makes a red and white flower. Hopefully that idea is pretty simple. And we see again an example of that here, where in incomplete dominance, if you cross that, instead of getting, you would expect 3 red, 1 white, but you don't get that. Instead, you get 1 completely dominant, which makes it red, 1 completely dominant recessive, that makes it white, and 2 pink, which is that blended phenotype. In codominance, again, you would get 1 that is completely homozygous for red, 1 completely homozygous for white, and 2 mixed, but instead of being pink, they're red and white because it's codominant. So now we're given this practice question about incomplete dominance here, and we're told that we have coat color, where if it's homozygous for B, that makes the coat black. If it's homozygous for W, it's white, and we have this blended phenotype of B and W making gray. So we're told that if you cross BW with BW, right? We get 25% black, 50% gray, 25% white. Now, based on that, what would happen if you crossed black with white? And what kind of inheritance is that? Well, we can literally do a cross for this, right? And I'll just write this out. If you cross BB with WW, you would literally get 100% of the offspring being BW. So the answer choices here are a little bit messed up, but we know that it is incomplete dominance, and we also know that you would have a phenotypic ratio of 1 that is gray and white. So the answer choice is B. And then here is a little chart I made about the different patterns of inheritance, starting with Mendelian and going to the different types that we learned. The idea here, I would just memorize, hey, what is the pattern we see in this? And how many genes are we working with, right? Because with Mendelian inheritance, we're either working with one gene in a monohybrid setting or two in a dihybrid setting. And also, here are some examples of them. That might come up on the test. For example, I've seen what type of inheritance is blood type. Well, it's multiple allelism, but it follows Mendelian inheritance of this distinct ratio. So I would, again, just try and commit this chart to memory. We see here a practice question based on this, and you can use the chart to answer this, but I don't know. Now might be a good time to practice this active recall skills, right? So we have a range of phenotypes. Well, that's already ringing a bell in my mind. It even says bell-shaped distribution right there, right? Influenced by more than 2 alleles, and the phenotypic expression is affected by dominant and recessive alleles, and there's an additive effect. There's a lot going on in this question, but you should have gotten it or had an idea as soon as it said bell-shaped distribution, right? Because when we hear bell-shaped distribution, we know, hey, something's going on here that's contributing to a range of phenotypes, aka polygenic inheritance, where multiple things are contributing to it. So we made it. That was a review of the first 4 lectures covering the basis of inheritance. Until next time, best of luck on your studies.